Welcome back, guys. We're looking at two new plates. We're looking at Macconkey Agar and EMB plates. And these two plates we'll find have a lot to do with one another. They're very similar plates in the way they're applied. We have learned about complex media, selective media, and differential media. In the case of the Macconkey Agar, it is both a selective and a differential plate. Quick review, that means that selective, it's going to kill some organisms while leaving others alone. And differential, those that grow will show different abilities. Maybe a color change versus no color change, things like that, right? Just like we saw with the MSA plate. So, first things first, let's take a look at what a Macconkey Agar looks like. Here we have kind of a reddish color again, okay? Not quite as bright colored as the MSA, not as opaque as the blood agar plate, but still kind of a reddish color. And this is an uninoculated plate, so nothing has been put on this plate over on the left. Next thing, we want to always know what are the key ingredients to these plates. We have, in this particular plate, bile salts and crystal violet. These two ingredients will inhibit the growth of certain organisms. Specifically, they will inhibit gram-positive organisms from growing. Okay? This is what will make this plate selective. All right? So the bile salts and crystal violet will inhibit gram-positive growth. This is quite the opposite of what we saw with PEA plates, right? PEA plates were the other way around, all right? We also see lactose, which is a sugar, which is an energy source, and we see something called neutral red. Not phenol red, but neutral red. All right. Now, if we were to plate a gram-positive organism onto the Macconkey Agar, and we know that there are ingredients in here that inhibit gram-positives from growing, we would expect to see exactly what you see up here in the top right. If there's no growth, it's a gram-positive. So you can see the etchings here. Somebody did a streak plate on here and then came back to see what happened, and there's nothing, right? So this would be a gram-positive organism. If this is the result, we say gram-positive and we're done. All right, but there are other possibilities. All right, down at the bottom, we see some growth on two different plates here, and not only do we see growth, which would indicate gram-negative, right? Because there is growth. They were not inhibited, so we have to say they were both gram-negative. But we also see a difference in the way these organisms are growing. So we have to take a closer look, right? Well, you definitely see color to the growth, so that's important to recognize. That's good scientific observation. But not only do I see color around here on the bottom left, I see color associated with the actual colonies, kind of a pink-purple color associated with these colonies. They also look kind of mucusy, right? Kind of gross. They have kind of probably some capsules there that make them look that way. But over here on the bottom right, yes, I see kind of a bright color, but it's even more red and more vibrant. And on top of that, you can see all throughout the medium, there is red around the colonies, okay? Which is indicative of a precipitant that has formed, okay? This precipitant is kind of a red color there in the medium. If there's color to the colonies, like you see over on the left, it's a lactose fermenter. Color almost always means that there was some kind of fermentation, some kind of breakdown going on that caused a change in pH. In this case, neutral red is going to show us this change in pH. It's not going to change yellow like phenol red does. It's actually going to show a brighter kind of pinkish purple color like you see here in this image. So if you see color to the actual growth, it's a lactose fermenter. Now over on the right here, where we're saying not only is there color to the growth, which means there's a lactose fermenter, but it's such a strong fermentation process that the sugar byproducts, when this sugar is broken down, are really acidic and cause these precipitants within the medium, which tells us it's a really strong lactose fermenter. Now in this case, right here on the bottom right, we're looking at E. coli. E. coli is a classic strong lactose fermenter, and it happens to be one of the things that we might be looking for when we use a Macconkey agar. Macconkey agar is applied to look for fecal contaminants, or sometimes they'll say coliform because it's colon, right? Or enteric for intestines. These are all terms that are used for some kind of fecal contaminant. So this E. coli, sometimes maybe you're looking in water to see if water has been contaminated or if some kind of food item has been contaminated. Any kind of E. coli on those things that are going to be consumed are not acceptable, all right, because they cause disease. All right, let's see. Let's look at, this is even a trickier aspect of Macconkey agar. So again, I wanted to show you this uninoculated plate, right? Kind of a reddish color, not very opaque, but a little bit darker with it than MSA. And here we have two organisms on the left and right. They're growing on this Macconkey agar. And the one thing that I want to look for is the growth a pink or purple color? Now on the right, it's certainly not the case. This is not pink or purple color. It looks like something else, right? And it's some tan or off-white color. Now over on the left, you could be tricked, right? It's not really obvious. It seems like this color of this growth over on the left kind of melds in, kind of blends in with the medium color itself. And when it does that, it is not convincing enough. It's not pink enough and it's not purple enough. These would both be considered technically a colorless growth. Even though you could argue with me that there's a little bit of color there, but it's not intense enough. It's not a nice purple or pink color. These are growing on the plate, so we know it's gram-negative. They weren't selected against, but they are non-lactose fermenters. In order for something to change the pH in the medium and change that pH indicator color, you're not going to have, you're not going to see that kind of thing. So non-lactose fermenters here. So I thought I'd show you one more with Macconkey agar. Here we've got three different ones growing, right? Here we have our E. coli. Clearly, there's color to the medium, right? That you see here kind of in this squiggle pattern. And there's absolutely color to the growth and color to the medium. And when that's the case, we're talking this bottom selection here, right? This has got to be something like E. coli. Now the next one down here, I'll circle it in red. This particular one has growth, so it has to be gram-negative, but it's definitely not a pink-purple color. So this would be a colorless growth. Gram-negative, non-lactose fermenter. Okay, we'll switch colors to, let's go to a purple color here. Down at the bottom, we have, I see some color. All right, I can see some color here. It's not as convincing as the one at the top, but this tells me there is pink-purple color going on. It is growing, so we would say this is a gram-negative lactose fermenter. We don't have on this particular plate an organism that was placed there and didn't grow, so we don't have a gram-positive organism on this plate. They're all gram-negatives. All right, so let's see. Anything else I want to tell you guys about this particular plate? I think that's about it. All right, the next plate and last plate for this particular clip has a lot of similarities to the Macconkey Agar. This is the Usin Methylene Blue. A lot of times they'll stick with EMB plate to name this, right? Because for the E, M, and B for short. And again, here we have kind of a reddish color, a little bit darker, kind of reddish brownish color going on with an EMB plate. This is an uninoculated plate over here. Okay. Now this plate has, like I say, a lot of similarities to Macconkey. It is selective and differential. It selects against gram-positive organisms. So this plate up here, where you see the etchings and yet nothing grew, this is a gram-positive organism for sure. Okay, I hope it's purple in our gram-negative, I hope it's purple in our gram stain, right? Now down below, I see that there is definite growth, and I see some pretty obvious colors going on with the growth down here in these plates. All right. Now before we really dive into that, let's not skip what is in these plates. Again, similar to the Macconkey Agar, we have lactose. That's our sugar source, that's our energy source. We also have eosin and methylene blue dyes within the plate. These are inhibited. They're going to be inhibiting gram-positive growth. So that's why gram-positives can't grow. But these organisms clearly do, and on the left we see not only a gram-negative organism because it grew, but we see kind of a color to the growth itself. So just like in the Macconkey Agar, if there's color to the growth, we know there's fermentation. And in this case, it's similar again to the Macconkey because it's lactose in the plate. So it's lactose fermentation going on. Now, if it grows gram-negative, and it's a very strong lactose fermenter, like we know E. coli is, then it will even form really kind of a dark black a lot of times color and a green metallic sheen on the surface. Kind of beautiful, right? I mean, we don't think of E. coli, something that can be pretty nasty depending on where it ends up in our body, as a beautiful thing, but I think I'd like to have a car with that color, right? The green metallic sheen is pretty cool. Now, let's take a look at the non-lactose fermenters. These are gram-negative organisms. They are not inhibited by these dyes we talked about, but they clearly are not taking on the dark purple or black with metallic sheen, all these crazy colors. There are again kind of questionable colors going on here. Okay? And so when they're kind of questionable colors and they're not really vibrant, well, they're most likely going to be non-lactose fermenters in that particular sense. So I wanted to throw this this one at you. Let's look at this one on the top. So this organism, there was Micrococcus is the name of the organism, which it's not really my concern that you remember every name. I'll mention that now. I'm not really concerned you remember every name of every organism that does or doesn't grow on these plates. I mean, E. coli is a big one, But beyond that, I'm not really concerned. In this case, this organism was plated on an EMB plate, and nothing grew at all. So what do you think that is? If it doesn't grow, this is an EMB plate, no growth means gram-positive, right? Because gram-positives cannot grow. So we know that this is a gram-positive organism, said and done. And we can't say anything about whether it can ferment lactose at this point, right? If it was killed because of the dyes that are in this plate, we can't really elaborate as to whether it can, in fact, ferment anything. All right? But down here, let's look at the bottom plate. Now this plate is tricky, right? Because if I look carefully, I see some white kind of colorless, definitely not black or purple, doesn't have a green sheen to it. So these particular colonies look to be more colorless than anything. They're not really taking on the purple or dark with a green sheen, these colors we'd expect from lactose fermenters. So this is actually a mixed culture down here on this plate. I can tell because we have, use purple for purple, right? Because I can see here some purple colonies. So this indicates to me that there is pink-purple growth, gram-negative organisms that can lactose ferment, and it indicates to me with those that are kind of an off-white color that this is more of a colorless growth, no pink or purple going on, and these are gram-negative non-lactose fermenters. So we have actually 2 organisms growing on that particular plate. All right, so I think I'll leave it at that, and we'll jump into some other plates here. We're almost done. We only have a couple more plates and media to go over. Okay.